They're ready in Linden. Okay, so thank you again for letting me present from Prosser. We have a big crowd here also, growers who are participating. So what I'm going to talk about now is the blueberry coldhardiness and a project we've been doing that's been funded by the Blueberry Commission. And I really think about it as the business and science of growing blueberries. So if you're not familiar with coldhardiness and mitigation in Eastern Washington, high bush blueberries are primarily in winter hardy from -14 to 20 Fahrenheit. But the spring and the fall can be a problem. And even sometimes midwinter hardiness, we'd worried about, well, are you truly hardy at that point or not? So people spend a lot of money just on propane alone. This doesn't, these figures don't account for time, labor, hours of lost sleep, decrease from your life expectancy of watching blueberries temperatures. So a lot of times we think that probably looking at dormancy and measuring it has been fairly easy or straightforward, but it's a little bit of a more complex issue. And I thought if you're not familiar with some of the things that go behind dormancy and the science behind it, I'd fill you in on these next 3 slides. So there's really 3 types of dormancy, and this is from a horticultural standpoint, which makes this really an interesting project, is that there's para, endo, and ectodormancy. And affecting us most of the time is endodormancy is when it begins from the inside with plant hormones. And ectodormancy begins with external environmental cues. So you get this, almost the exact opposite of a coldhardiness problem is seen in California, where they have all these models looking at chilling units and whether or not they got enough chilling days. So what we have though here is we get cold weather, shorter days. That's when the plant starts to go through ectodormancy and it says, oh, okay, well, the environment's acting upon me. I need to go to sleep, and I go into ectodormancy. But then enough chilling units have occurred, and there's always some nice week in February that occurs, and the plants say, woo, I'm ready to go. And hormones start to come through the plant, and they really go through endodormancy at that point. But you can get bad weather, and you can get times where you go back and forth between this endodormancy and ectodormancy, and it really creates this interesting complex interaction between plant science and the environment. And it's that we're trying to model and that you all are trying to control. So there are different stages of dormancy. There's acclimation, which if you look on this graph, I'm going to use an arrow, and I'm hoping the arrow comes through on the other side of the state. But on the graph, as you look at it, as it starts to decrease, that's when plants are acclimating to the cold weather. They are becoming more cold hardy. You have deep dormancy in the bottom of the graph, and that's the middle of winter. And then plants start to come out and they deacclimate, which is when on the right side of the graph, they start to slope upwards. So all plants follow this curve. How fast they slope down, how fast they slope up, really depends on the genetics, the preceding temperatures, and very often the phenological stage. So we have seen in the literature, and then actually I'm going to show you some examples of where we see it again, where in deep dormancy you can reacclimate. So a plant we can start to get some warmer weather in the middle of winter, and a plant can reacclimate to say if it gets colder that it can sustain deeper temperatures. But how much it does that will depend on if it's past bud swell or not, and which cultivar you have. Okay, so now let's look at how cells freeze. And I'll tell you a little bit, relate it back to blueberries. So this is a slide Lisa DeVetter has used for her class. It's really good. It tells you that there's really 2 types of freezing that can occur in a plant. And maybe you'll think of this winter as you're out there with your plants. So ice is dry. And if you get this extracellular freezing, so this is on the left, you get ice that forms in that tannish bar on the outside. What happens is on the inside of a cell, it's mainly made up of water, and the water moves to where it's drier. When you do that, you dehydrate this cell. The cell basically it dies from dehydration. And why blueberries are particularly sensitive to this anyway is that they acclimate to the cold by dehydrating themselves in the first place. So less water means that they'll have less water in them, means that they'll have less to freeze. The other way that cells can acclimate themselves is more like how grapes do it. Grapes, or how you might do it while you put salt on the ground or on your driveway, is that grapes will bring sugars and other carbohydrates into their cell, and that will lower the freezing temperature. And it doesn't really reduce its actual water content. The other way that freeze injury can occur is that you can get ice crystals that form inside the cell, and it's a direct damage where the cells actually rupture and burst. Okay, so we know there's a need, we know there's some complexity, but we are also conducting research, so I'm going to show you how we do it. And show you what we have now is this graph or chart on the left. And it's really the chart that we use is from Michigan State. And it goes by each stage, which has a good scientific validity that, you know, for each stage, bud swell, green tip, etc., there is a set temperature in which the buds will freeze at. So this one says bud swell, it's good to 10 to 15 Fahrenheit. So it isn't done here, and it doesn't account for preceding temperatures or weather, and it doesn't account for cultivar. So it is the best that we have, but it doesn't refine you into any precision agriculture. Where we need to be in is more in line with what they have for grapes. So the grape industry had the advantage of looking at this for 30 plus years. They were able to make a model of it, and that model is posted on Ag WeatherNet. And so it looks basically, the web page looks like this. So you can open up the model, you can select your Ag WeatherNet station closest to your field, you can select the year, and you can select the cultivar. And then out will come a graph and a model, and it will, because it's Ag WeatherNet, it easily plots the maximum temperatures and the minimum temperatures in a day. And then the very bottom red, yellow lines are the hardiness temperatures. Those hardiness temperatures were actually developed and known because of years of research where they cut open buds and looked at them, or did DTA analysis and said, so at these temperatures, what will happen with the buds? How will the preceding temperatures occur, etc. But because that research, they're able to then predict, use a model. Okay, so this is what we do. We go into the lab and we have a freezer, and on the west side of the state, Lisa DeVetter uses a glycol bath. We have a freezer. It looks a little different than this now, but this is a better picture, more exciting than what we have now. So what we have is, we have basically on the east side of the state a programmable freezer, or a programmable system that allows us to set to slowly freeze buds or canes and to slowly thaw them. And that is more indicative of what happens in nature, but it also prevents us from having false negatives, so, you know, or false positives. Once they have gone through this freeze chamber and we pull them out, we do 10 different temperatures every single week. And once they've gone through the freeze chambers, we let them sit for a day. They can oxidize. And then we have crews on both sides of the state that meticulously cut open the buds. And so this is a whole blueberry bud, but inside you have multiple flower initials. Each one of those flower initials will become a flower, and each one will become a blueberry, of course. but we count how many flower initials are within one bud. So we average about 8, but sometimes there's 10 or 4, etc. per bud. And we look at how many are dead and how many are alive. And we do it for a lot of buds. So it's a very time-consuming process. It takes, the cutting takes 2 days with 2 people. Okay, so we have a lot of key partners, people who will help us through this. And the very first one, I want to let you know, Washington Blueberry Commission has funded this work. We have cooperating growers on both sides of the state that bring us samples, keep us on track. And we also have collaborated with other people who are in doing cold hardiness work. So there's cold hardiness work going on in tree fruit and in grapes. And so we rely on them pretty heavily, and it's nice that all of that is simultaneously going on. But I just got done telling you that our methods can be time consuming. So we have spent some of our time trying to build a better mousetrap. And if we built a better mousetrap, could we ask different questions? So I work with engineers at the Center for Precision Ag and Automated Systems. In Prosser, I work with them on a lot of other sprayer projects that I talked about yesterday. And we also have engaged some horticulturists and modelers from, or David Jabot from Oregon State University, and he's looking at this exact same problem in cherries, and cherries has similar cold hardiness methods and plant strategies that blueberries do. So it gives us nice synergy for what's being learned in cherries. We can also learn and pursue and check to see if it's correct in blueberries. So what the engineers we've done is we've spent very nominal amount of money to say, are there different ways that we can use cameras, hyperspectral imaging, or this is micro CT or thermal cameras to look at water content, because we know that when a bud is dead, it will hold less water. So can we detect the percent mortality or different rates of mortality by using some type of imagery system? The straight CT has not had good success, neither has the thermal camera. We have tested initially the micro CT, which can basically get you down to a refinement of a couple centimeters. And micro CT is used a lot in medical equipment, and we'll probably pursue that a little bit more in the spring of this year because it's relatively cheap. The cost of overnight shipment and $30 for a technician to run it through the micro CT. The problem is it is still destructive. What we really wanted was a non-destructive, something I could take to the field with me and say, is there damage? If there not, then what happens horticulturally after that? If we tagged it, how much damage can you sustain and still have good fruit quality? And some of that is very much from the line of thought of cherries. So you can sustain some damage, but you get larger cherries when you don't have, when you haven't over cropped. So we just wanted to look at that. There are other potentials. So this hyperspectral imaging, we take the buds, we go through the same freeze process, and we take an image with a hyperspectral camera. There's a very complex analysis and set of steps that they go through. And we want to know at a certain wavelength, will the two pieces, will we be able to detect healthy and damaged? At the same time, we can do it the old-fashioned way of good horticulturists, and we can weigh things. So we know that every growth stage, we know this even from the Michigan charts, that every growth stage has different temperatures of how cold hardy they can be. And if we can more accurately determine where that plant is by just by how much water content it has in there, we could more accurately tell you what temperature it's going to be cold hardy at. So what we do with this is we weigh, do a fresh weight before it's we do a fresh weight of all these buds before they come into before we do anything to them. We dry them down for 2 days. We take a dry weight, and the difference between the two weight numbers is how much water was there. That's simple, basic science from high school. So we're using those numbers to continue building our model. So I want to show you some of our results because we've been doing this for a couple of years. And so I wanted to kind of summarize it over the past several years. So what we have learned, and we have moved along, changed some of our focus, but still good to go back from 2015 to 16. That's the winter. So December 2015 to spring of 2016. And then again, November of 2016 to spring of 2017. What we know with blueberries is that they acclimate, meaning the front part of the curve, when they go down, they acclimate very quickly. And doesn't matter what variety you are. Here in these, we had Aurora, Draper, Duke, and Liberty. They all acclimate very quickly. They are very cold hardy in the middle of winter. And then there's less varietal differences in the second year than the first, but we still see that Aurora and Liberty are slower in deacclimating in the spring. So what that means is that your cultivar difference, and when they come out, they're early to wake out of bed. Or they're slower to wake out of bed. I think I said that in reverse. I should go to bed more some days. So if you look at this in Mount Vernon, because now you want to see, is it the same, different? How is it run on one side of the state versus the other? So on that side of the state, we were looking at Duke and Draper, and both over, again, those same years. And Duke and Draper follow each other closely. That shouldn't be too much of a surprise because they have very close, their parentage and genetics is very closely related. Surprising to me, I thought that the winter cold hardiness for like midwinter at the lowest part of the curve would be less, but it is actually really cold hardy in the dead of winter. But it's when they're coming out that will differ between Eastern and Western Washington. And I'm going to show you that in the next slide. So this is East versus West, right? So hardiness, remember, is tightly linked with stage of the crop. And in Mount Vernon, which of course we know this to some extent. So Mount Vernon is the graph on the left. So they come into bud break 20 days ahead of Eastern Washington in Prosser. The problem with that is that you're not as cold hardy once you've hit bud break and beyond. So in some cases, if your temperatures are, if we're going to come through and we've already gone into bud break in early March, you are hoping that there's no cold snap after that. We can almost in Eastern Washington always have some kind of cold spell somewhere in the time of March. And so it's that timing that can sometimes be tricky. With that being said, Lisa and I early on in one of the first years, I want to give you this example, right? So we said, okay, so we have some of these curves. When would we have possibly ever had an issue in either the east or west side of the state? And this time the graph for Prosser is on the left and the graph for Mount Vernon is on the right. And so these curves are from 2016 data, right? So the curves took 1 year of data. They indicated temperature that the buds will experience 50% mortality, right? So this says that at the very beginning of the year, the buds were in dormant, or at the very beginning of our data set for Mount Vernon, they could have sustained somewhere about -3 degrees before they had 50% mortality. The snowflakes we went back and we said, okay, over the past 20 years, when would there have been incidences if all the curves were the same throughout the years, which they won't be, but you're looking at it. That's why I called the graph the hypothetical historical happenings. So, but when could they have experienced a problem? And in Eastern Washington, we have times in the early fall, and actually this is our early winter, sorry, we're in 2008, 9, and 13, where we could have had problems where the two where the temperatures got cold enough to cause damage. In 2014, coming out, we would have also may have seen some damage. And on the west side of the state, you have two incidences, one in 2008 and one in 2011. So for those of you who have really good memories and you say, I remember 2008 and we had no problems. So this is the hypothetical historical happening. This curve is not from 2008, it's from 2016, but it is to say, so in the dead of winter, when could you have had problems and would there ever be an incident? And that's sort of our best way to say, should we continue some of this work or not? Is it relevant? Is what we're finding relevant to continue? And just looking at historical data compared to with the data and the modeling we're doing now, I would say yes, because there's enough years on both sides of the state to say that there should have been some mitigation for cold climates. Okay, so to talk to you about what we're seeing in the hyperspectral, there's a lot of graphs here and there's a lot of information, and really what you need to know is this. Our greatest desire in these graphs, so these graphs represent wavelengths, and our greatest desire is to see a separation in the wavelengths. So the stronger the separation, it says, there's healthy buds, damaged buds. So can we pick out a distinct wavelength that would determine or indicate to us that we have damage from either 0 to 10, 31 to 60, or 61 to 100. These are not what you guys make decisions on, right? You would make decisions probably on much smaller classification units within this class 1, 2, and 3. But interestingly, in our models for grapes, if you can find the LT50, you can easily calculate what the lethal temperature at 50, where 50% of the buds die. You can easily calculate the lethal temperature or model at where 10% die and where 90% die. So what we really want to do is find a class here that gives us one strong separation. And the key to this, and the good results for us, are that in the dormant season, when you're probably worried about it the least, you have some separation in the lines. You can get some of the wavelengths, so we could develop a sensor that would be pretty good in that dormant season. As you go in the spring, where you guys are the riskiest time periods, from tight cluster, early pink, you get a much stronger separation, meaning that we have a greater chance of developing a very cheap field sensor that would just pick one wavelength, and it would be relatively inexpensive to go out and look at some horticultural experiments and maybe find a little easier to measure damage and death rather than cutting for 2 days. And so this is very promising. We're going to bring another student in who will complete this work and try and look at some field sensor technology as we go forward. Okay, so this is the part that you've been waiting for years, is the Coldhardiness model. And so this is an example of what we're trying to build and continue building from last winter's data or all the previous winter's data. And then we're going to take this winter's data and next winter's data, hopefully, if it's funded, next winter's data, to try and vet some of this. This is an example of Duke in 2 different locations in the eastern Washington. And this bottom line looks just like that grape graph I showed you. Here we have lethal temperatures at 10, 50, and 90 degrees. And the upper graphs, the upper squiggly lines, are your temperature. So you have temperature at the top, you have coldhardiness at the bottom. And you can see the idea is that never the 2 lines shall meet. That's what you want. So, but you can see, again, this really interesting thing that was reported in the literature, but it's within these red circles in both locations. We see reacclimation. So the grass has started to come up. The buds have started to have maybe some type of either endo or ectodormancy at that time. Said, yep, spring is coming. Some cold weather happened. You can see this snap right here. the buds reacclimated and became more cold hardy that same week, and then came back, started to get fairer weather. You see the weather started to go back up again, and the buds started to go back up. Right now in cherries, and we're not 100% sure yet, and I can't find anything determinative or definitive in the literature, but in cherries, your time period is really post bud swell. We don't see a lot of reacclimation, but even all the way through bud swell, you will see reacclimation occur. So I can't promise you that that's going to occur all the way for blueberries, but we definitely still see it during this time period. And this all occurred of these reacclimations this is our second year we've seen it, and it's been in that February time period for us. Okay, so those are results, but what are we going to do with it? At the end of this, what we'd really like to do is get it connected to all of you. AgWeatherNet is already used by growers. Some growers have told me on the eastern side of the state that they've already stopped midwinter fan treatments, which is appropriate because we really never get as cold hardy as what the temperatures are. Still that spring and fall temperatures that are the most concern. But prior to this experiment, I know, and even 2 years ago, I know I was doing a sprayer calibration class, and it was in the middle of winter, and they were telling me that they had turned all their fans on their blueberries. And it's that type of thing that can be very expensive if we're not sure how cold hardy those experiments are, or how cold hardy the blueberry plants are. So for us, our plan is after the model is vetted to get it incorporated into AgWeatherNet. We've already talked with them, and it should be available to you all in the next couple of years. Okay, with that, I can take any questions. If you have any, does anybody in Prosser have any questions to start with? Any questions here? We're a very quiet group here. Okay, Chris, I didn't see if you texted me any questions.
So we don't have any questions here, but I think we want to, at least from here, we want to thank you, Gwen. This was a great presentation, and look forward to seeing that integrated AgWeatherNet. So.
Okay. Thanks.
Yeah. Mute this. Okay, our next talk is going to be interactive. Wake everyone up. We're going to get to do another clicker survey. Suzette is going to start handing out clickers. Title of the talk is Biodegradable Plastic Mulches for Profitability in Agricultural Applications. And there's a presentation followed by the survey, right? Oh, as they're integrated. Okay, so we'll wait for everyone to get a clicker, but go ahead and take it away, Gwen.
Thanks. Hi, everyone. My name is Quan Chen from Washington State University. So here is my colleague, Suzette Garrirado. She's handing out the clickers. So for today's presentation, I'm going to presenting the overview of strawberry market and two types of mulches And the key details of our study on consumer willingness to pay for strawberries grown on biodegradable plastic